All right, the second part of Chapter 5, we are going to start talking about the transmission of information by neurons. And this starts on page 10 of the first PowerPoint. So, neurons have to transmit information, and they do it they have a unique ability to undergo rapid changes in electrical potential across the cell membrane. This is the key to a neuron's function. So they can undergo those rapid changes. The electrical potential across the membrane is the distribution of ions, which creates a difference in the electrical charge on each side of the cell membrane. So we talked a little bit in the electrical stem lecture last quarter about how if you have insulators separating spaces, you can have a capacitor, you can store charge, and that's exactly what the phospholipid membrane of the cell acts like an insulator. It prevents ions from crossing, and it maintains a gradient of ion concentration from the inside of the cell to the outside of the cell, and that's what creates that electrical potential. So if we didn't have that difference in electrical charge, we wouldn't have any chemical potential. So in order to rapidly change that electrical potential, we need to have membrane channels, because there needs to be a way for the ions to flow across the membrane. So we have these proteins that are embedded in the phospholipid membrane of the cell that allow ions to flow across the membrane. And there are 4 different types, and we'll talk about each of these individually. One of them, the leak channel, is passive. They're always open. And then 3 of them, modality-gated channels, ligand-gated channels, and voltage-gated channels, open in response to a stimulus and close when the stimulus is removed. So you can sort of think of the gated channels are the inside of the cell is like a gated community. The ions want to get in, and they have to have a key or a voice recognition or something in order to get into the gate. And as soon as they use their key, the gate closes again, and you remove the stimulus, and so the channels only open in response to that stimulus. The leak channels, they are always open, so a small number of ions are leaking through at a slow, continuous rate. And I think of it as like a drip irrigation system, where there's a little bit of water dripping out through holes in the hose into your garden. Instead of a bunch of water rushing out at one time, there's a continuous small leak. And those leak channels, they help maintain the electrical gradient. They keep the osmotic gradient going. Just by letting a small amount of sodium or a small amount of potassium through, they maintain those electrical gradients. So gated membrane channels open in response to specific stimuli. So the modality-gated channels, they open in response to mechanical forces, temperature changes, or chemicals. So there's sensory changes like stretch, touch, pressure, temperature. That is like all of the things that we did in modalities lab last quarter. Soft tissue mobilization, thermotherapy, cryotherapy, those are all mechanical forces or temperature changes. So those open those modality-gated channels. Ligand-gated channels, ligand means bind, so they open in response to a neurotransmitter binding to the surface of a channel receptor on the postsynaptic cell membrane. So you need the binding of that neurotransmitter in order to open the channel. So it's like the neurotransmitter is the key, and it fits in the keyhole of the ligand-gated channel, which opens the door to let the ions go through. Voltage-gated channels open in response to changes in the electrical potential across the cell membrane, and those open and close almost instantaneously. They're important in the formation of action potentials and the release of neurotransmitters. So we'll talk more about how the voltage change propagates down the neuron. But those voltage-gated channels are really important in that propagation of the membrane. So a modality-gated channel a lot of times starts a response. Voltage-gated channels propagate it, and ligand-gated channels happen at the synapses in order to propagate to the next neuron. So they all sort of work together to propagate a nerve impulse. So electrical potential is a difference in electrical charge, in that electrical charge is carried by ions, so sodium and potassium. So in pathology, when we were talking about the consequences of fluid imbalance and electrolyte imbalance, that is going to affect our nerve conduction. So that's a big deal, right? It's really going to affect our electrical gradients, right? So the difference in positive and negative charge inside and outside the cell membrane oppose each other to help control ion movements. So that unequal distribution of ions is what creates the difference in electrical charge on each side of the membrane. So rapid change in the electrical charge across the cell membrane transmits the information along the length of an axon. And then when you gets to the presynaptic terminal, it elicits the release of chemical transmitters to other neurons or other electrically excitable cells, like the membrane of a muscle or an endocrine gland. So there are 3 types of electrical potentials in neurons that are essential for transmitting information: resting membrane potential, local potential, and action potential. And we're going to talk about each one of those individually. So the resting membrane potential is the value of the electrical potential across the membrane when the neuron is not transmitting information. That's why it's called resting. So the difference in positive and negative charges inside and outside the membrane, because we have negative ions that are trapped inside the cell, and they actually, those big negative ions that are trapped inside the cell can't go through the channels because they're huge. So the ones that can go across back and forth are the smaller ones, the sodium and potassium. We have a steady state condition at rest with no net flow of ions across the membrane. So at the resting membrane potential has no net change in the total distribution of ions across the 2 sides. Although individual ions can move across through leak channels, that is really just to maintain the chemical gradient. There is not a lot of, there's no net change. So little sodiums are leaking into the cell, little potassiums are leaking out of the cell, but overall there's no net change. And that's why it's the resting membrane potential. So at rest, there is the cell membrane serves as a capacitor, like I said earlier, separating the electrical charges on either side of the plasma membrane. And the unequal distribution of the ionic charge across the membrane is essential for the neurons to be excitable. So if the ions could just willy-nilly travel across the membrane anytime they wanted, there would be no electrical chemical gradient, and the neuron would not be excitable. Something would change and it wouldn't matter because you would be at equilibrium and there would be no change in the charge. So that electrochemical gradient is hugely important for maintaining the excitability of the neurons. So 2 forces determine the ion distribution across the plasma membrane. The concentration gradient, so concentration of the individual ions, and the electrical gradient. So opposing chemical and electrical forces control the movement of ions. Equilibrium occurs when there's no net movement of that ion across the membrane. So the resting membrane potential is measured by comparing the electrical difference between the inside and the outside of the cell membrane. At rest, the inside of the cell membrane is measurable at approximately minus 70 millivolts. So it's 70 millivolts more negative than the outside of the cell membrane. Okay, and you will see some sources that say 75 or 80 millivolts, but when you think about how tiny that is, it's in that range. So inside the cell, the resting membrane potential is maintained by passive diffusion of ions across the cell membrane, so just those leak channels, and active transport of sodium and potassium by sodium-potassium pumps. So a sodium-potassium pump for each cycle, which takes one molecule of ATP, it actively carries 2 potassium into the cell and 3 sodium out of the cell. So sodium and potassium are both positively charged ions. So now we're getting less potassium than sodium, so less positive in the cell, more negative in the cell. That's where we get that 70 millivolts more negative inside the cell than outside the cell. So those ATP-run sodium-potassium pumps actively help maintain the unequal distribution of sodium and potassium to maintain that resting membrane potential. So the concentrations of sodium and chloride are higher on the outside of the cell compared with the inside of the cell, whereas the concentration of potassium is higher on the inside of the cell compared with the outside of the cell. So chloride is a negative ion, sodium is positive, and potassium is positive. High concentration of unneutralized negatively charged molecules, which we're calling anions, inside the cell, those are those big fat ones that can't get out, they also contribute to the negative resting membrane potential. So in the diagrams in the book, they indicate the anions as an A with a little minus sign. So it's a negative charge in the resting membrane potential. When we look at this, the anions are those big fat purple ones inside the cell. The sodium are the little red ones, the potassium are the little green ones, and the chloride are the little blue ones. So the distribution of those ions helps maintain the negative resting membrane potential. So the electrochemical gradient in neurons and membrane resting potential are maintained by negatively charged molecules, those anions, trapped inside the neuron that are too large to diffuse through the channels. So it keeps sort of a baseline negative charge on the inside of the cell. Passive diffusion of ions through leak channels in the cell membrane, and active pumping of sodium and potassium across the membrane. All three of those things maintain the resting potential. So if we didn't maintain that resting potential, if we just had open channels all the time, we would have no electrochemical gradient and no potential for excitability in the neurons. So that's a big deal. The sodium-potassium pump uses energy from ATP to move ions across the membrane against their electrochemical gradient. So normally, potassium wants to move out of the cell and sodium wants to move into the cell. The pump goes against the gradient and moves potassium into the cell and sodium out of the cell. So 2 potassium into the cell and 3 sodium out of the cell with each cycle. So we're setting up a negative potential inside the cell. As long as the cell has ATP, we can maintain that unequal distribution of potassium and sodium across the membrane. So ATP is important. We have to keep that electrochemical gradient in order for neurons to be able to do their magic. So we get an alteration in membrane potential when ion channels open to allow selectively the passage of specific ions. So when we talk about those different types of channels, that's where we start getting a change. That's where we start changing the potential. So the changes from the resting membrane potential, we get sudden brief changes in membrane potential where the membrane can become depolarized or hyperpolarized. So depolarized is where the potential becomes less negative than the resting potential. Hyperpolarized is where it becomes more negative than the resting potential. When the membrane is depolarized, it increases the likelihood that the neuron is going to generate a transmittable electrical signal. So that's an excitatory state when the membrane is depolarized. When the membrane is hyperpolarized, the potential becomes more negative than the resting potential. That is an inhibitory state because it decreases the neuron's ability to generate an electric signal. So that's important. An excitatory impulse is one that's going to propagate potentially down the neuron. An inhibitory impulse is one that's going to prevent propagation of the impulse down the neuron. So a lot of, we'll talk about, when we talk about neurotransmitters, whether they're excitatory or inhibitory, that's important for control of what's going to happen in the nervous system. So gradual, longer-lasting changes in membrane potential can also occur. So a lot of times we have these fast, transient changes, but you can have gradual, longer-lasting changes, which is called modulation. So there's small changes in the membrane's electrical potential that alter the flow of ions across the cell membrane. And we will talk more about modulation in Chapter 6. But that is, know that there are small changes that are kind of the normal change from resting membrane potential. But with modulation, we can get longer lasting changes.